Hey Bio 102, let's get to it. Most of this video will likely be a review from previous coursework or from Bio 101. But let's ease into cell bio. So we're all made up of cells, which is the smallest unit of life. And since we're all animals, we are made up of animal cells. So here's a diagram of an animal cell to the right. Animal cells are a type of eukaryote, which I will define in a moment. And our body contains 200 different variations of this animal cell type, and they vary greatly in terms of their structure and function. So for example, our red blood cells, they actually do not contain a lot of these organelles being shown here. So in 101, you talked a lot about atoms and molecules and even macromolecules. In 102, we will talk about molecules, macromolecules, and cells, and the organelles that make up the cells and cells that make up tissues. And in 103, you'll talk a lot about cells, tissues, and organs, and then make up organ systems, which then make up organisms, which you'll talk about in 104. You'll talk about the evolution of organisms and how organisms interact in an ecosystem. This is the trajectory that you are on. Now from 101, I hope you can appreciate some of the universal features of all living organisms. First of all, they store their genome in the form of double-stranded DNA, which can be replicated. They transcribe portions of their DNA into RNA molecules. They use proteins as catalysts. They translate RNA into protein in a similar way. Each protein is encoded by a specific gene, and they require a continual input of energy. They are enclosed by a plasma membrane. And life, because it's so ordered, is anti-entropy. The reason there are these universal features to all living organisms of living cells is because we arose from a common ancestor cell. And that common ancestor cell then evolved and diversified thereafter. So many of these features of that common ancestor cell that were crucial to its survival have been sustained over billions of years. So for example, importantly, a consequence of that, the genome of one cell is readable by other types of cells in other organisms. So you can take a piece of DNA from a human cell and put that into a bacterium, or a piece of bacterial DNA and put that into a human cell, and that information will be successfully read, interpreted, and copied in those cells. So this demonstrates the universal processes of transcription and translation, which you talked about in 101. So all life on Earth is related, and therefore they have fundamental similarities. Our planet is populated by living things, which we call organisms, and they are each essentially chemical factories that take in matter from their surroundings and use these raw materials to generate copies of themselves. Right? And these organisms, as you will appreciate in 104, if you don't already, are extremely diverse. So here we have a colony of unicellular bacteria, a butterfly, a rose, and a dolphin. And what do they all have in common? Well, they're all alive, right? So how do we define a living organism? How do we define life? So in your previous coursework, if you covered this, life may have been defined in abstract general terms related to growth, reproduction, and an ability to actively alter their behavior in response to their environment that they are in. Now that we know more about cells, however, we can talk about this in terms of cellular life, as all living organisms are composed of cells. So these cells are the smallest unit of an organism that abide by these three characteristics of growth, self-replication, and the ability to respond to environmental stimuli. So the cells of all of these living organisms here, the bacteria, the dolphin, and the flower, they are fundamentally similar on the inside, and they resemble each other in regards to their chemistry. They're composed of the same sorts of molecules, which participate in the same types of chemical reactions. They all have a genome, which contains genes written in the same chemical code, and they're constructed out of the same chemical building blocks, and they're interpreted by essentially the same chemical machinery. Right, the RNA polymerases and the ribosomes, and they're replicated in the same way by DNA polymerases when a cell duplicates itself. Therefore, we can perhaps now think of this fundamental view of life from the view of a cell. As all living organisms are fundamentally made up of cells, each of these cells possess fundamentally similar chemistry, and operate according to the same basic principles that you discussed in 101. So the bottom line here again, because again, all living organisms are made up of cells and they are fundamentally very similar. So this brings us to the central dogma, right? So hopefully you remember this from 101. This is universal to all living cells on Earth. Genetic information flows from DNA to RNA in a process called transcription, and from RNA to protein in a process called translation, and that's the central dogma. And it is the special relationship between DNA to RNA to protein that makes this self-replication of cells possible. Just take a minute and think about this question. So pause the video. Are viruses alive? Are they living things? How are they different from cells? Hopefully you've had a moment to pause and think about that, right? While viruses do contain genetic information in the form of either DNA or RNA, they do not have the ability to reproduce themselves by their own efforts, right? Instead, they have to infect a cell, take over their machinery to reproduce themselves. But viruses are truly not living. They are merely chemical zombies in a way. They are inert and inactive outside of their host cell, but they are able to then exert control once they gain entry into the cell to replicate themselves. So what is a cell? It is the minimal self-reproducing unit of life. They pass on a copy of their DNA to progeny cells, which then allows it to then form the structure and all the proteins necessary to make a functional cell, which can then self-replicate. They consist of self-replicating collection of molecular catalysts encoded by their own DNA. They are enclosed by a membrane. They are driven by free energy, and they're released in a complicated network of chemical reactions. When comparing cells, one of the most obvious places to start is with size and shape. So here we have two extreme examples. So this bacterial cell on the left is typically a few micrometers in length. So each of those Cheeto-looking rods is one cell. On the other extreme, we have on the right a frog cell, a frog egg, okay, which you'll talk about in 103 a bit. Each of those frog eggs is a single cell, and it has a diameter of 1 millimeter. So that's actually, you can see that with your naked eye. So if you do the math there, this frog egg on the right is hundreds, if not 1,000 times larger than a bacterial cell. So enormous range of size. They also have different shapes. Also note the different size scales here of these different cells. In A, we see a drawing of a single nerve cell from a brain of a mammal. This cell has a single unbranched extension called an axon that's projecting towards the top of the image. And it is through that axon that it sends electrical signals to other nerve cells. In B, we see a paramecium, which is a single giant cell that dwells in ponds, and it is a unicellular organism. So it exists as a single cell. And it's shaped like a submarine, and it swims by means of these beating cilia, which are these hair-like projections that cover its surface, and they beat in a synchronous manner to allow for it to move forward, just like a swimmer performing the breaststroke. In C, we see the surface of a flower petal, which displays this orderly array of tightly packed cells. So these cells are immobile, and they have cell walls composed of cellulose, which you may recall from 101 is a carbohydrate that can form rigid structures. In D, we see a macrophage that has spread or flattened itself out. So this type of macrophage is highly mobile, and it can crawl through tissues, constantly transforming its shape to squeeze into tight spaces while it patrols that tissue. In E, we see a fission yeast. It's shaped like a rod, but it's caught in the act of dividing into two, which is a process called mitosis. And we see that this yeast has been labeled with fluorescence, the red denoting something called the medial septum, which is forming a wall between the two nuclei, which are also stained in red. And the green here is the cell membrane. From this panel of images here, we can really appreciate the diversity of cells in their sizes and their shapes, and ultimately, thus, their function. Okay, so if all living organisms are made up of cells and the cell itself is the smallest unit of life, it makes sense then on the evolutionary time scale that the first living organism would have been a single-celled or unicellular organism. And then later organisms made up of many cells together, multicellular, would then have evolved. So before humans, before mammals, before dinosaurs, before multicellular organisms, there were microbes or unicellular organisms. So when did the first cellular living organism arise, if you had to guess? So the first known single-celled organism appeared on Earth about 3.5 billion years ago. And that's roughly about 1 billion years after the Earth had formed. So life on Earth had humble beginnings as a single cell, and more complex forms of life then emerged later on. And the first multicellular organisms not appearing until only about 600 million years ago. And while we will focus on eukaryotes in this class, and in 103 and in 104 you will focus on multicellular organisms composed of eukaryotic cells, we should importantly note that unicellular organisms on Earth certainly outnumber the number of multicellular organisms on Earth. And also pretty amazing to consider, I think, if you consider the unicellular bacteria in your gut outnumber the eukaryotic cells of your entire body. So you are technically more bacterial cell than you are eukaryotic cell. And if you expand that out to all life on Earth, not only does unicellular life outnumber multicellular life, but it is also much more diverse. So stepping back a moment to review something that you learned in 101 that's related to this, right? The complete DNA sequence of an organism defines its nature in exhaustive detail, right? And so we can use genomic analyses. That's a powerful tool to simply and directly determine evolutionary relationships between organisms. So after sequencing the genome, you get a string of letters, the nucleotides, the A's, C's, T's, and G's, as you can see in the background of this image on the right. So that string of letters can be entered into a computer and compared with the corresponding information from other organisms. And because DNA is subject to random changes that accumulate over long periods of time, the number of differences between the DNA sequences of two organisms provides a direct, objective, and quantifiable indication of the evolutionary distance between them. That is how we can establish our evolutionary relationships and how we can conclude that we are more related to a mouse than we are to a fish. So to really drive home this point, here's just a little practice activity. So this would be an example of what you might expect in lecture as a part of that lecture participation, right? I'll give you some sort of question like this. You'll write your answer. You'll write your name in your section letter. So take a moment, read over this question, pause the video, think about it. I'll have the answer on the next slide. So try it out before you see the answer. Analyses such as that allow us to identify the three branches of life on Earth: the eukaryotes, the bacteria, and the archaea, each of which has unique properties. While prokaryotes may be less familiar to us, eukaryotes, on the other hand, make up the life that is most familiar to us, like plants, animals, fungi. So what is the difference between prokaryote and eukaryote? Well, eukaryotes possess an organelle called the nucleus, while prokaryotes do not. And you will see that viruses are nowhere to be found on this tree of life, but they, as we are very well aware of, ubiquitous. Viruses are not living organisms, but they are biological entities, and they have evolved to prey on living cells. Bacteria have a very simple structure, and they come closest to showing us life stripped down to the essentials. It contains no organelles other than ribosomes. Sometimes you'll see ribosomes classified as an organelle, even though they're technically not. That's just more of a historical thing. But they do not have a nucleus to hold their DNA. They are also small. They generally are just a few micrometers long, although there are some giant species. Prokaryotes often have a tough protective coat, or something called a cell wall, surrounding the plasma membrane. So in addition to their plasma membrane, they often have a cell wall. Most prokaryotes live as single-celled organisms, although some are capable of forming chains or clusters, such as these cyanobacteria here. Eukaryotes, on the other hand, are bigger and much more elaborate. Some do live independent lives as single-celled organisms, such as this paramecium. Many eukaryotes live in multicellular assemblies, so they are multicellular organisms. In this course, we will focus on eukaryotes. And on the next slide, I will show you a black and white image called an electron micrograph that's really zoomed in on the subcellular components of a eukaryotic cell. So to help orient you to this strange new landscape of the cell's interior, imagine we're slicing this cell here right down the equator. So imagine, for example, you are cutting a tomato, right? You're cutting a slice of tomato and you're going to slap that tomato slice on a microscope slide on a plate, and now you're going to look down at that tomato slice. So this is one of our first electron micrographs in this class, and you will see a lot of them. And in this micrograph, or image taken with a microscope, you can make out some prominent eukaryotic organelles. And what's an organelle? It's a compartment. So maybe take a minute and pause the video, try to identify or make out whatever organelles you have learned about before, and don't worry if you can't identify any right now or don't know any organelles of the cell yet, that's fine. What you can appreciate is that there are different compartments, and you can actually distinguish between them because they have these boundaries in darker gray, right? So they're enclosed by a membrane. And we can see that some are large and round, and some are bean-shaped, and some are spaghetti noodle-shaped. And also importantly from this image, we can really appreciate that the cell is very jam-packed. It's a very busy space. So let's start with this biggest one. What organelle is this? Well, that is the nucleus. That's the nucleus of a eukaryotic cell. So again, by definition, eukaryotic cells keep almost all of their DNA in the membrane-enclosed nucleus. And that's usually the most conspicuous organelle, or the easiest one to pick out. So we have the DNA here. In the nucleus, it's packaged with protein to form chromosomes, which typically only become visible under in the light microscope when they condense in preparation for cell division or mitosis. The nuclear envelope is actually a double membrane, and that surrounds the nucleus, and it separates that nuclear DNA from the cytoplasm, which in the eukaryotic cell includes everything between the plasma membrane and the nucleus. Here's an expansive list of other differences between prokaryotes and eukaryotes in the conventional classification, being the nucleus, absence or presence. Prokaryotes tend to be smaller than eukaryotes, and that can be appreciated in this image here. These bean-shaped organelles called the mitochondria are comparable in size to the average bacteria. And keep that in mind. That's important. Other differences include how their genomes are structured. Prokaryotes typically have a circular genome that's not packed into chromosomes with histones. They also tend to have one copy of their genome, so there's something called haploid, whereas our eukaryotic cells are diploid. We have two copies of the genome. There are other differences as well, which we will not focus on, but their mechanism of cell duplication is different. Their outer membranes have different compositions. Eukaryotes have lots of other organelles, such as that bean-shaped mitochondria. And we'll talk about those organelles, which is why I have that highlighted. They also have slightly different ribosomes, that important machinery involved in translation. So the ones in blue here are the ones that we will focus on and the ones that I would like you to remember. But there are plenty of other differences. So bottom line here, eukaryotic cells are generally bigger and more elaborate than bacteria and archaeal cells, and their genomes are bigger and more elaborate too. And that greater cell size is importantly accompanied by all these intracellular organelles as well. Even amongst eukaryotic cells, we can see similarities and yet differences. And actually, this is the key to biology of living things. So if I were to wrap up the entire biosequence in one sentence, it would be this: living things share similarities yet are strikingly diverse. So the whole of biology is this counterpoint between these two themes: astonishing variety in individual particulars and astonishing consistency in fundamental mechanisms. Here we have two eukaryotic cells. We have an animal cell and a plant cell. We see a nucleus in each. We also see other organelles present, but yet we can appreciate differences between the two, right? So just by eye, we can appreciate that. So what contributes to the diversity that we see in the living world? Well, the cellular diversity can be attributed to either differences in the genome between different organisms, they have different genomes, or it can be attributed to the differences in genome expression between different cell types of the same organism. So what do I mean by that? So let's start off with the scenario on the left. Right here are the same two images I showed you on the previous slide, an animal cell and a plant cell. The plant cell and the animal cell, they contain different genomes, and therefore they have different instructions for how to make all the proteins present in that cell, and therefore their function will be different. And the processes carried out in those cells will be different. But we also can appreciate immense diversity within one multicellular organism, right? So for example, the cell types of the human body. Here, we are looking at a different type of electron microscopy, where instead of looking at a slice of the cell, we're looking at the exterior of the cell. And what we're looking at here are 3 different cell types of the blood. There's the red blood cell, the platelet in the middle, and the white blood cell on the furthest right. And they are all human cells. They're all from human blood. So they contain the same genome, yet they look very different, right? So how can that be? And you'll talk about this a lot in 103, but this is due to differences in how that genome is read or interpreted, such that some part of the genome might be read or expressed more often in one cell type than in a different cell type. And that allows multicellular organisms, such as ourselves, to have different cell types, each with a different structure and function. And that's going to contribute to the collective functionality of our body. On that note, multicellular organisms, they're made up of lots of cells, but these cells exist in tissues. So here we have, on the left, we have plant tissue. So we're looking at the root tip of a fern, and the DNA containing nuclei are stained in red, and they are surrounded by a thin cell wall in blue, light blue. And then on the right, we see cells of the intestinal crypt. The nuclei are stained in blue. So ultimately, let's just have some review. Cells are compartmentally organized. They possess a genome and a means by which to use it. They're capable of making more of themselves. They acquire and use energy. They carry out chemical reactions. They sense and respond to stimuli. They are capable of self-regulation. They engage in mechanical activities, right? They move and squeeze and transform their shape, or some do, much more of that than others. And importantly, they evolve. So to end this pre-lecture video, here are your key concepts. Cells are the smallest denominator of life. Single cells and unicellular organisms are invisible to the unaided eye. We'll need microscopes, right? All cells are made from the same classes of biomolecules and are fundamentally similar. All life on Earth is related. There's a root to the tree of life. And we started out unicellular, and then multicellularity evolved later. All life on Earth is characterized as prokaryote or eukaryote. Prokaryotes have cytoplasmic genomes. Their genomes are not enclosed by a nucleus. Eukaryotes have nuclear enclosed genomes, and they also have other membrane enclosed organelles. Though they are small and possess similar basic chemistry, cells have evolved into a vast variety of shapes and sizes and functions, right? That similarity, yet diversity. So I'll see you in class Bio 102.